નીટ ની ડેટ આવી છે બેટા, સાંભળ્યું હશે તે 12 સપ્ટેમ્બર, બરાબર ને?
હા સર.
પેટર્ન માં ચેન્જ છે, ખ્યાલ આવ્યો કે એકવાર સમજાવવું એ શું છે પેટર્ન માં?
નો સર.
ઓકે, તો હું તને એકવાર સમજાવું છું. આ વખતે પેટર્નમાં આ લોકોએ ચેન્જ કર્યો છે એટલે થોડું કેરફુલ રહેવું પડશે. એટલે એમાં બેનિફિટ એ થઈ શકે છે, અમુક વસ્તુ તમે અમુક ચેપ્ટર ઓપ્શનમાં નીકળી શકે છે. હવે જો બેટા, આ પેટર્ન આ લોકોએ કરી છે, બરાબર? એટલે શું કર્યું છે તમને કે સેક્શન એ બી માં ડિવાઈડ કરી દીધા છે. એટલે સપોઝ ફિઝિક્સ છે તો એમાં સેક્શન એ બી કરી નાખ્યું, કેમિસ્ટ્રીમાં બી સેક્શન એ બી કરી નાખ્યું. હવે સેક્શન એ માં 35 ક્વેશ્ચન હશે ફિઝિક્સ અને કેમિસ્ટ્રીમાં, બરાબર? હવે આ લોકોએ બાયોલોજીને બી શું કર્યું છે કે બોટની ઝૂલોજી, કેમ કે એમસીઆઈ શું છે આપણા આખા સિલેબસ ને બોટની ને ઝૂલોજીમાં ડિવાઈડ કરે છે. આઈડિયલી તો ઘણી બધી બ્રાન્ચીસના ટોપિક છે, પણ હવે કોઈ કારણસર એ લોકો વર્ષોથી આવી રીતે ડિવાઈડ કરે છે. એટલે આમાં ખરેખર જોવા જઈએ તો આની અંદર બી એ લોકો એવી રીતના ઓપ્શન આપે છે 35 અને 15, 35 અને 15. એટલે સેક્શન એ જે હશે, બરાબર, એમાં 35 હશે બોટની ઝૂલોજી બંનેમાં, અને બી માં 15 હશે. હવે આ બધામાં બી તમે જોવો તો 15 જ છે, એટલે આમાંથી ગમે તે 10 કરવાના. ફિઝિક્સ કેમિસ્ટ્રીમાં 15, આ 35 તો કમ્પલસરી કરવાના જ છે. ફિઝિક્સ કેમિસ્ટ્રીમાં 35 તો કરવાના જ છે. બોટની ઝૂલોજીમાં પણ 35 તો કરવાના જ છે. આ ગ્રીન જે કર્યા એ કરવાના, એ સોરી, આ જે કર્યા ને આપણે, સપોઝ હું તને ટીક કરીને ખ્યાલ આવે એટલા માટે કહું છું કે હું જેટલા ગ્રીન થી ટીક કરું છું ને કમ્પલસરી જ છે. આ કમ્પલસરી જ છે બેટા. હવે આ જે બીજા 15 વધ્યા ને તમારે બધામાં તારે ફિઝિક્સ, કેમિસ્ટ્રી, બોટની, ઝૂલોજી એ બધામાંથી આપણે 35 તો કમ્પલસરી કરવાના જ છે. એટલે કેટલા થાય? 35 into 4, 35 into 4 થયા ને કમ્પલસરી કરવાના. અરે એક જ મિનિટ આ બેટા,
યસ સર.
એક જ મિનિટ. ક્યાં જતું? ઓકે, એટલે ટોટલ શું થશે કે 140 ક્વેશ્ચન્સ તો કરવાના જ છે, બરાબર? એટલે 140 into 4 કરીએ આપણે, એટલું તો આપણે કમ્પલસરી અટેન્ડ જે છે કરવાનું જ છે. ઓકે, એટલે 560 માર્ક્સ નું તો કમ્પલસરી કરવાનું જ છે આપણે. હવે આ જે 15 દરેકમાં જે વધ્યા છે એમાંથી 15 માંથી વિલ ગેટ ઓપ્શન્સ. તો ફ્રોમ ધેટ યુ હેવ ટુ ઓનલી એટેમ્પ્ટ 10, 10, 10, 10. Okay, એટલે from 15 you have to section B માં you have to attempt 10 questions from the given 15. એટલે you'll get options. Limited option છે, but it is good anyway. So અમુક chapters એ depend કરે કયા ટોપિક ના પૂછે છે હજી. So 10 into 4, 40 questions થઈ ગયા બેટા. ઓકે, અરે સોરી 40 માર્ક. હા, ઓકે, તમારા 40 questions થઈ ગયા. એટલે 40 questions છે,
40 marks.
એટલે 40 into 4 160 માર્ક્સ થઈ ગયા ને, બરાબર? એટલે હવે
Yes
સી
sir.
આમાં શું સિનારીઓ છે કે દરેકની અંદર as you are attempting, just a moment, physics chemistry માં શું છે કે physics માં out of 15 you have to attempt 10, chemistry also out of 15 you have to attempt 10. Now biology as we have two subjects as such. Okay, so because of that bioma, that is you will get two options. See 15 બતાડ્યા છે. Botany, zoology you'll get. So out of 30 you have to attempt 20 questions. એટલે આ 40 થયા. So 40 into 4 is 160 marks જે છે. એટલે આમાંથી you have to attempt 15, 15, 30. Okay, અને આમાંથી 20. તો આઈડિયલી જોવા જઈએ તો બાયોલોજીમાં 30 ક્વેશ્ચન્સ હશે, એમાંથી 20 એટેમ્પ્ટ કરવાના છે, અને આમાંથી 10 કરવાના છે. એટલે 160 માર્ક્સ જે છે આવી રીતે થાય છે. તો ટોટલ શું થયું? આ 560 પ્લસ 160 કરીએ આપણે તો 720 થઈ ગયા ને બેટા?
720.
Everything remains the same, only you will get options that in section B you will get option for each of this, from where you have to attempt only 10 questions. ખ્યાલ આવ્યો બેટા?
Yes, sir.
એટલે આ રીતે પેટર્ન ચેન્જ કરી છે. નાઉ ઇટ વી આર નોટ શ્યોર કે આમાં અગેન આ જે ઓપ્શન્સ આપશે એમાંથી વોટ they will do. એટલે કદાચ there are two ways to look at it, કે કોવિડ સિચ્યુએશન ના લીધે કદાચ થોડું ઈઝી કરવા માટે બી કર્યું છે આ, અને અનધર વે ઇઝ કે મેરિટ બહુ અપ જાય છે તો એના માટે બી કદાચ એવું કર્યું હોય. તો પછી એવું બની શકે કે આ જે 10 ક્વેશ્ચન્સ જે છે અને આ 20 ક્વેશ્ચન્સ જે છે એનું ડિફિકલ્ટી લેવલ જે છે એ વધારે હોય. બરાબર, એવું બની શકે કદાચ, બટ વી આર નોટ શ્યોર. ઓકે, કે શું હશે. મોસ્ટ પ્રોબેબલી એટલું ડિફિકલ્ટ ના બી કરે બીકોઝ ઓફ ધીસ કોવિડ સિચ્યુએશન, બટ મેરિટ જે રીતે અપ જાય છે એ રીતે કદાચ ડિફિકલ્ટ કરી નાખે આ ક્વેશ્ચન્સ. ઓકે બેટા, તો આ પેટર્ન છે કે વી શુડ નો વોટ વી આર ગોઈંગ ટુ અપિયર. હવે કોઈ આમાં તને કોઈ પ્રોબ્લેમ હોય તો કે, એઝ યુઝ્યુઅલ બાકી બધું સેમ જ છે, નેગેટિવ માર્કિંગ ને બધું.
No, sir.
ઓકે, તો હવે શું કરવું છે આપણે? આઈ થિંક આપણે શું પૂરું કર્યું હતું?
Evolution complete.
તો હવે તારે શું કરવું છે?
Sir, biomolecules.
ઓકે, બાયોમોલેક્યુલ બેટા મેં ઓલરેડી મારી ચેનલ પર બધા લેક્ચર મૂક્યા છે એના, સો બેટર કે એને બહુ હ્યુજ ચેપ્ટર છે, તેમાં 8-10 લેક્ચર છે જે થશે, તો બેટર કે તું એમાંથી એકવાર જોઈ લે, પછી તને સ્પેસિફિક ડાઉટ હોય એ કરાવી દો.
Okay, sir.
મેં ઓલરેડી બાયોમોલેક્યુલ્સ ના મેજોરિટી લેક્ચર કે હ્યુજ ચેપ્ટર છે, તેમાં અગેન છતાં બી તારે કરવું હોય તો હું તને ટેક્સ્ટ બુક બેઝ કરાવી દઉં અમુક વસ્તુ ને, કેમ કે એમાં ફર્સ્ટ ચેપ્ટર જ હું સ્ટાર્ટ કરું છું. એમાં મેઇનલી ઇમ્પોર્ટન્ટ છે ન્યુક્લિક એસિડ, પ્રોટીન્સ, બરાબર, અને પછી એન્ઝાઇમ નો પાર્ટ. તો હું તને એ કરાવી દઉં એવું હોય તો એમાંથી.
Okay, sir.
બાકી તો ઇફ યુ રીડ તને આઈડિયા આવશે. ઓકે, અને કેમ કે ઓલમોસ્ટ એમ સમજ કે 10 to 12 લેક્ચર્સ થાય છે બાયોમોલેક્યુલ્સ માં મારે, એ ઘણું હ્યુજ ચેપ્ટર છે એટલા માટે. તો બાકીનું તને ટેક્સ્ટ બુક માંથી જસ્ટ કહી દઉં છું, પછી યુ કેન ગો થ્રુ. ઓકે, અને આઈ થિંક લેટ મી ચેક મેં કયા કયા ઓલમોસ્ટ મેં લગભગ 5-6 લેક્ચર્સ તો મૂક્યા છે. સોરી, ગુજરાતી મીડિયમના મૂક્યા છે. ઇંગ્લિશ મીડિયમના આઈ થિંક એનું મેં 1 કે 2 લેક્ચર મૂક્યા છે. મારી પાસે પડ્યા છે લગભગ શોર્ટ કરેલા, તો પુટ ઇટ એવું હશે. એનીવેઝ વિલ ટેક ઇમ્પોર્ટન્ટ ટોપિક્સ, ઓકે, એઝ યુઝ્યુઅલ, ને પછી આઈ એક્સપ્લેન.
Okay, sir.
Okay, now first part what they are doing is they are basically in this part or here they have given the method. Okay, or here they mentioned the method to analyze the macro and micromolecules. Okay, so how to perform the chemical analysis? So it is very simple. You add a living tissue and then you add it to, I mean, you basically add the trichloroacetic acid, then you basically you completely crush it and you obtain a thick slurry. Now then you isolate or then you filter this slurry. So the thing which remains on the filter is retentate and the thing which will pass is what we call as the filtrate. So the thing which will pass will contain acid soluble fraction, while the thing which will be on the filter is acid insoluble. It's like when we prepare the tea leaves will be on the filter. That is what we call as retentate. The tea which we drink is filtrate. This acid soluble pool has so many organic compounds. Okay, so if you analyze the cell, then the cell will contain different organic compounds, and depending upon their size and all, they will show this sort of soluble compounds in the filtrate and insoluble in the retentate. Okay. Also, in the acid soluble pool, you can see inorganic compounds like sulfate, phosphate can also be seen in that. Okay. Now this question they may ask. That's why I mean taught it over here. Now this is in relation to something which has been mentioned over here, that is macro and micromolecules. Okay, so all of these soluble acid soluble compounds are usually have molecular weight in these ranges. Okay, now basically there are two types of biomolecules, that is what we call as biomicromolecule. Okay, so there are mainly four types of major biomolecules which is found inside the cell or inside the living body. Those are proteins, nucleic acids, polysaccharides, and lipids. Okay, now except lipid, all are macromolecules. Except lipid, remember that all are macromolecules. So all have molecular weight more than 10,000 Dalton. So usually micromolecule will have molecular weight ranging from 18 Dalton to 800 Dalton. Ideally, it is 1000 as per chemistry. And macromolecules will have molecular weight more than 800 Dalton. So proteins, carbohydrates, and nucleic acids are macromolecules. But lipids are in this range, so they are micromolecules. Okay. Now remember, all of these are found in acid insoluble fraction. જેટલા પણ proteins, lipids, carbohydrates છે, these are all found in acid insoluble fraction. Ideally, acid insoluble will have macromolecules, but lipid is an exception because it is a size-wise lipid is macromolecule, but it comes under acid soluble fraction. So be clear with where you will find these compounds. They may ask this question. Okay. So basically, if you analyze the same thing for a single cell, then the cytoplasmic composition will show acid soluble pool, while the macromolecules will be seen in the acid insoluble fraction. Okay, nowadays we do not use such technique. We rely on chromatography and all. So make sure you are clear with this, and if you have any problems, you can ask me from this. તો આનું રિલેશન ત્યાં આગળના ટોપિક સાથે છે, એટલે મેં એવી રીતે કરાવ્યું. Now, other method which they have given is basically over here. It is to obtain the inorganic compound. So take a living tissue, you dry out, so H2O will go out, then you burn it completely, so you'll get the ash, and ash will contain all these inorganic elements. Now we are talking about we'll discuss four main biomolecules in this chapter, that is proteins, lipids, nucleic acids, and carbohydrates. Most important are protein, sorry, and then we have nucleic acid. So we are starting with the protein. So protein are mainly, they are made up of what we call as polypeptide. Proteins are what we call as, they are made up of polypeptide. So these amino acids are linked to one another and they form this polypeptide. Okay. Now, if we talk about the general structure of amino acid, then they have a central carbon which is termed as alpha carbon. It is attached to the R group. Then it has amino group. Okay, also it has carboxyl group and a hydrogen attached to it. So as you know, this is also termed as chiral carbon. In chemistry you might have learned. So this is alpha carbon. This R group is variable group and it is different in each and every amino acid. So we have 20 different amino acids and 20 different amino acids, 20 different R groups. That is what they have mentioned. Okay. Now, if R group is only hydrogen, we refer to that as glycine. If it is only methyl group, it is alanine. If it is hydroxyl methyl, it is serine. Okay. So if you see over here, this you should know. There's only instead of R group, everything remains the same. See, amino group is there, hydrogen is there, carboxyl group is there. Only what changes? This R group. This is R group. Glycine is also termed as the simplest amino acid because instead of R group, it is hydrogen. So this is also referred as simplest amino acid. Then there is alanine. This is methyl group. Then there is methyl hydroxyl group in this area. So we can classify amino acids on the various types as such. Okay. So it can be either based on the number of amino and carboxyl group. They can be acidic, like glutamine. You remember these examples, huh? But 9 examples are there. They're asking this from textbook. Basic is lysine, neutral is valine. Okay. Then amino acids can also be aromatic, like there is tyrosine, phenylalanine, tryptophan. And they have, as we know, ionizable nature. That is, they have amino and carboxyl groups. So in solutions of different pH, the structure of amino acid changes. The structure of amino acid changes. So there is one important property which we are talking about is the zwitterion form. Now basically you should know that amino acids are amphoteric compound. Amino acids are amphoteric compound. Amphoteric means they have properties of both, that is acid and base. Why? Because carboxyl group will give acidic property, amino group will give basic property. Okay, so what will happen that this carboxyl group, okay, it can act as a hydrogen donor because it is acidic, or proton donor you can call it, while the amino group, okay, while the amino group can act as a proton acceptor or H plus acceptor because it is basic. So now at certain pH, what will happen? That each of these amino acid will exhibit certain, both the charges. They'll exist as dipolar ion. They'll exist as dipolar ion. So this is what we call as zwitterion. This is what we call as zwitterion. So amino will show positive as it will accept. CEO will basically donate, so it is referred to as negative charge. So they'll ask you this question, that which of the following is zwitterion? So you should know that જ્યાં બંને charge દેખાય, wherever you see two charges, okay, those are zwitterion. And zwitterion are seen at specific pH. So for each amino acid, this pH will be different. So for each amino acid, this pH will be different, and we refer to that pH as isoelectric pH. We refer to that pH as isoelectric pH or PI value, also called as isoelectric point. तो જે pH પર zwitterion exist કરે એને PI value કે isoelectric pH કહેવાય, અને એ pH પર zwitterion બંને charge show કરશે. So whatever they give you, wherever you see two charges, it is zwitterion form. Carboxyl will show negative, amino will show positive, and we refer to this form as the zwitterion form. And this is the peculiar property of the amino acids. So for 20 amino acids, each one will have separate PI value. So for 20 amino acids, each one will have separate PI value, and the reason is because of the characteristics of the carboxyl and amino group, which we have discussed. So this is what we call as zwitterion form. So આમાં કોઈ problem છે બેટા? રાઈટ, આવે છે અવાજ બેટા?
સર
ક્યાંથી ડિસ્કનેક્ટ થઈ દીધું બેટા?
નેટવર્ક ઇશ્યુ.
અરે નહીં, કયા ટોપિક થી એમ કહું છું?
These Twitter RNA.
આખો ટોપિક મિસ થઈ ગયો છે કે મને ખ્યાલ નહોતો એટલે?
No sir, iso electronic और वो सब हुआ.
ઓકે ઓકે. Okay, so see, for each amino acid, Twitter ion or the pH at which they will exhibit Twitter ion form will be different. So for alanine it will be different, for serine it will be different. Okay, so for each amino acid this isoelectric point will be different, and it is because of the carboxyl and amino group which they have. So it is very important and you should know what it is. So આમાં કોઈ doubt હોય તો બોલ બેટા Twitter ion.
No sir.
Okay, now see, the other part of the proteins had been mentioned over here because we take everything together. So basically proteins are polypeptides, and they are, as I told you, the two amino acids which are linked to one another, and that is they are linked by peptide bond. They are linked by peptide bond. So each protein is a polymer of amino acid. It has 20 different types as we have discussed. So protein is a heteropolymer because each amino acid is different. Okay, now there are various ways where you can classify amino acids. So one of the most important ways which has been mentioned over here, that is these amino acids can be essential and can be non-essential. So if we get amino acids through our diet, okay, then they are termed as essential amino acids. Like when we eat dal, legumes, okay, all those sources which are rich in protein, then we get amino acids. Those are essential. Okay, remember the essential. And the one which is manufactured by our body, okay, from the other raw material or other biomolecules, they are non-essential. So as such, you can classify 20 amino acids in essential and non-essential ones. So this is also definition is important. Apart from that, over here they have mentioned the structure of the proteins. So there are 4 levels of structure, that is primary, secondary, tertiary and quaternary. So it is on the base of their polypeptide chain shape. It depends on the shape of their polypeptide chain. So it is very much useful in the chemical configuration and the chemical studies of the proteins. So if it is a straight line, we refer as primary structure. If it is a helical arrangement, we refer as secondary structure. Or if it is this sort of arrangement, also we refer as the secondary structure. If it is a highly complicated structure, we refer as the tertiary. And 2 or more tertiary can unite and join quaternary. 2 or more tertiary can unite and join quaternary structure. So the primary structure, you can obtain information about the which amino acid will be first and which will be last. So you can get an information about the sequence of the amino acid in a polypeptide. So it is the simplest structure. Primary structure is the simplest structure, and here you imagine protein as a line, as you can see over here. So always at the left end there will be first amino acid, at the right end there will be last amino acid. This is not as such functional. Primary structure, the protein chain polypeptide which have been folded in the primary structure, it is not usually functional. Okay, so this is primary structure. As you can see, it is like a straight line. So this and left end you have first amino acid, and over here you have last amino acid. So the first is termed as N terminal, the last is termed as C terminal amino acid. It is termed as C-terminal amino acid. So this is what we call as primary structure. So the thread is folded in the form of this helix. The thread is folded in the form of the helix in the secondary structure, where you can see the helical arrangement is there. All it can show such helix is termed as alpha helix. It is termed as alpha helices or alpha helix. It is right on. It is turned towards the right-handed side. Or if the protein chain is in this form, that is what we call as the beta sheet. Or if the protein chain is in this form, that is what we call as beta sheet. Okay. So this is also secondary structure. These both are what we call as secondary structure. So if it is in helical form, like a revolving staircase like this, okay, or like this beta sheet structure, we call it secondary. Only right-handed helices can be observed. If you know about our hair contains keratin protein. So keratin protein has alpha helical structure. Keratin protein has alpha helical structure. So remember the example of alpha helix is keratin, which is the common hair and nail protein. So over here you can see two polypeptide chains are there. Here also you can see this is one and this is another one. Two chains are there in beta sheet also. So secondary structure has shapes in the form of helix and beta sheet. Then we have more complex is tertiary. Okay, so tertiary structure, let me show you something else, just a moment. We'll have these two bonds mainly in the amino acids, that is hydrogen and disulfide bond. Okay, so this is biologically, this is the most functional protein, and it is mainly found in abundance in the bodies of the living organisms, which is what we call as tertiary structure, and it gives the three-dimensional view of the polypeptide chain. Then we have quaternary structure, which has, as you can see, it has two or more this is the hemoglobin example. Hemoglobin has 4 subunits, that is 2 alpha chains and 2 beta chains. So we refer to hemoglobin as alpha 2, beta 2. This is alpha 1, alpha 2. This is beta 1, beta 2. So it is more than 2 tertiary structure which are connected. So it is one of the most complex structure. Like adult hemoglobin has 4 subunits, 2 are identical. So this is what we refer as this. They asked this MCQ, what does alpha 2 beta 2 means? The answer is human adult hemoglobin. Infant hemoglobin will have alpha 2 gamma 2. Infant hemoglobin will have alpha 2 gamma 2. As we grow, the gamma chains are replaced by the beta chains. The gamma chains are replaced by the beta chains. So I'll just show you something in relation to this. મેં તને બાયોમોલેક્યુલ્સ ની નોટ્સ પણ મોકલી છે બેટા, તો you can also go through that.
Okay, sir.
બહુ ટાઈમ પહેલા મોકલી હતી, just go through them, દરેક ચેપ્ટર. So see, they asked you this question in 2019 that To determine the structure of proteins, what can be used? So answer is Ramchandran plot. Okay, so if you where the unit starts, okay, they've given some introduction of different scientists in your textbook. So this was given by G. Ramchandran. He was an Indian scientist. Okay, I think he was from University of Madras, and he gave this. He was basically he had a background in physics, and then he gave this plot or a graph through which you can determine what amino acids are there in the polypeptide. Okay, so and this graph looks something like this. The graph looks and something like this. So you can get to know what structure it has. So they'll not ask you in detail because it comes at in the post-graduation level. So as such, you should know that it is determined, it is used to know the structure of amino acids because this question was asked in 2019 NEET. NEET or AIMS, I'm not sure, but it was asked. Okay, so you should be aware about this. These are four structures as I have shown you, as you have discussed. Primary structure protein is usually seen as a straight line. Okay, so peptide bond can be seen. Protein is not functional. Okay, so the newly formed proteins on the ribosomes, okay, will exhibit this sort of structure, which is formed on the endoplasmic reticulum, will exhibit this sort of structure. It is unstable structure. Then beta sheet we have, as you can see, zigzag information or the helical structure, as you can see. Polypeptide is folded to form alpha helix or beta sheet. As you can see, it is spirally helically coiled structure. The bonds which you see are hydrogen bonds. Examples are myosin, keratin, tropomyosin. Keratin is important, so I had mentioned that. Beta sheet, other example is fibrin, which is the silk thread protein. So you can see two polypeptides which are parallel or anti-parallel, and then they form this sort of structure like this or like this. So you can see H bond also in them mainly. Tertiary structure, you have example is enzymes which will study. So protein looks like a polypeptide looks like a woolen ball form. So on the periphery region over here, you will see hydrophilic amino acids. On the inside, you will see hydrophobic. Okay, because the peripheral part will come in contact with the water. The bonds you see are hydrogen bond, disulfide bonds mainly in this structure. Disulfide bond is between sulfur containing amino acids, that is methionine cysteine, and it is most important for biological activity. So it is biologically active form. It gives the 3D view also. The example of tertiary structure is globular proteins. The example is globular proteins. So majority of enzymes and proteins in our body or in protoplasm inside the cell are in tertiary structure. They are in tertiary structure. The bonds which you see are peptide bond, hydrogen bond, disulfide bond, hydrophobic bond, ionic bond. Quaternary structure, there are two or more peptide polypeptides are there which can be similar or dissimilar. So it can be either in this cube shape or in this linear string of sphere shape. So this both the shapes can be seen in the quaternary structure. Over here in our textbook, it has been mentioned about the hemoglobin, where you can see these two structures, alpha 2, beta 2. Okay, this is alpha 1, alpha 2. Blue one is this alpha, and the red is beta for polypeptide. So 2 alpha, which refer and 2 beta. In infants, it will be alpha 2 gamma 2 gamma are replaced by beta chains in the adults. This is also a question of AIMS. This is the real hemoglobin structure. As you can see in the RBC, we have it has heme group, that is iron containing group. This is iron part. So iron is very much necessary for the hemoglobin functioning. You can say this is alpha chain, this is beta chain. Okay, so this is what we have in the protein structure. Got it, beta?
Yes, sir.
I had sent already. I had sent you okay long way back. So check out. You can go through that. Then if you want to classify proteins, okay. So as you can see, this is image from the old group. This is the secondary structure. If you focus on this part, you'll get this sort of amino acids, different amino acids linked. Like this is serine, this is cysteine, tyrosine, glutamic acid. So each amino acid can be shown by 3-letter symbol or by 1-letter symbol. Like serine is SCR and S, cysteine is CYSNC, tyrosine is TYR and Y, glutamic acid is GLUNE. So you should know this, okay? Those are in earlier textbook, which was used being till 2018. And these are all these serine, cysteine, tyrosine, and glutamic acid. Now, when we classify proteins, they can be simple, conjugated, or derived. Simple are globular or fibrous, that is, there's globulin, albumin, histone, or globular histone. We have covered keratin, collagen are fibrous. Conjugated have simple protein combined to prosthetic group, which is not, which is something non-protein, which is bound to this simple protein. Like we have glycoproteins, lipoproteins, lipids bound to proteins, sugars bound to protein, nucleic acids bound to protein. Like histone is an example of nucleoprotein. Milk protein is casein is an example of phosphoprotein. Hemoglobin is an example of the metalloprotein. Okay, so the cytochrome is an example of chromoprotein, for example. And protein can act as transporter, antibodies, hormones, enzymes, receptors. Like Rubisco collagen, you might know. The most abundant protein in the animal world is collagen. The most abundant protein in the entire world in the plants is Rubisco. Okay, that is the enzyme. Now they were asked in NEET 2019 this question about the GLUT, that is glucose transporter molecule. So in your textbook in the table, they will mention GLUT4. So if you know about the insulin, then this is GLUT. They will ask which of the following is insulin dependent glucose transporter. So if insulin is present, then this GLUT4 can transport glucose inside the cell. So the GLUT4 protein transporter molecule, which is found in the cell membrane, is termed as insulin dependent glucose transporter. Is termed as insulin dependent glucose transporter. While these 1, 2, and 3 are termed as insulin independent glucose transporter. This 1, 2, and 3 are termed as insulin independent glucose transporter. Okay, because they will not transport glucose on the basis of the presence of insulin, while GLUT4 will always transfer on the basis of the presence of insulin. So this was asked, and you should know it is in relation to endocrine system. But GLUT, for example, is a transport protein which is responsible for transferring glucose inside the cells, had been mentioned in this chapter. These are all important topics from proteins. Okay, so from textbook, just a moment, but I'm getting a call, so I have to attend.
Okay, sir.
Okay, right. So just a moment. Sorry. So see, we have covered about this is an important table from textbook, and GLUT4 question was asked from over here. This is insulin dependent glucose transport. So these are different protein they have mentioned over here. Rubisco, I hope you know, so I am not going in detail. Okay, better. So this is all we have in proteins. So proteins is very important for levels of structures and everything. So if you have any problems in proteins, you are free to ask.
No, sir.
Okay. Now we are discussing about the lipids. So lipids are generally water insoluble. Now from the lipids mainly, you should know that there can be either fatty acids or they can be either fatty acids bound to the glycerol. So you can call them as monoglyceride, diglyceride, or triglyceride. So they can be also termed as fats and oils on the base of their melting point. Oils have lower melting point, and they remain as oil inventors. So if you have seen, parachute will get freeze in the winter. Otherwise, the groundnut oil will not freeze. The parachute से hair oil जामी जैसे groundnut oil नहीं जामे. Why? Because it depends on their melting point. It also depends on the saturated fatty acid content. So saturated fatty acid વધારે હશે, that is, you can see water saturated and water unsaturated over here, then it will freeze. If it is less in amount, then it will not freeze. So groundnut or the edible oil will not freeze, while the coconut oil will freeze. Okay, then they've given important example of the conjugated lipids, like we have phospholipids, where lipids are bound to the phosphorus. They are found in cell membrane. One such example is lecithin. Lecithin is mainly found in the alveoli in the basement membrane, where it acts as a surfactant. So it is responsible for the exchange of respiratory gases in the lungs. Then we have neural tissue like neurons, which has myelin sheath. So that is made up of the sphingomyelin, or what we call as the sphingosine. They're also lipids. This is important for lipids. So you can see the structures of this palmitic acid 16, this phospholipid, glycerol, simplest lipid which we have, triglycerides, and cholesterol. Okay, so this may be asked, like identify the structure. Then from the polysaccharides, you know these carbohydrates are made up of the polysaccharides, and each polysaccharide has a many monosaccharide, has many monosaccharides. So we have examples of the different structure and different polysaccharides of glucose. There is cellulose, okay, then there is starch, and then there is glycogen. So all are glucose polysaccharide, but the structure is different. Okay. So you should know about all of this. Cellulose, starch has amylose and amylopectin. Glycogen is also a glucose polymer. Then there is inulin, which is a polymer of fructose. Fructose is a monosaccharide, as you know, commonly what we call as fruit sugar. So this is the glycogen structure part, which we have shown over the branch structure. These are all glucose units, and they are joined by glycosidic bond. They are joined by glycosidic bond. They are joined by glycosidic bond. Then they have given some examples of complex polysaccharides, like there are amino sugars, which is amine sugars bound to amino group, like there is glucosamine and acetyl-galactosamine. These are found in the exoskeletons of arthropods, okay, like cockroach and all. Their exoskeleton is made up of this glucosamine. So this question they'll be asked. So what we call as chitin. Chitin is mainly glucosamine. It's amino sugar, or what we refer as the complex polysaccharide. And these are mostly homopolymers. So from this glucosamine is important. Cellulose, starch, and glycogen, all are polymers of the glucose. That is important. Inulin is important. Also, they ask you this question. If you in chemistry, there is a normal test that if you add iodine to the starch, okay, it will give blue color because of the branch chain. But cellulose will not give blue color because it is not a branch, it's a linear structure. So if you add starch and iodine, you will get blue or violet color. While the same thing if you add to the cellulose, you will not get blue or violet color. Okay, so you should know that. So this is all important from the lipids and polysaccharides. Now we'll talk about the most important part, that is nucleic acids. Let me share just a moment. Right, chapter મોટું છે એટલા માટે આપણે સ્પીડમાં જઈએ છીએ. So if you have any problems, then you can ask, okay? But otherwise, I'm just focusing on what is important.
Okay, sir.
So see, now we are talking about the nucleic acids. Okay. So see, in nucleic acids, mainly the common examples which we take, okay, which we are going to study over here are DNA and RNA. So they are polynucleotides. These nucleic acids are made up of the polymer of nucleotide. Each nucleotide can have nitrogen base, a 5-carbon pentose sugar, and a phosphate group. If you remove the phosphate group, you will get nucleoside, that is nitrogen base and 5-carbon pentose sugar. So it is also very important. We also covered about it in the 12th. The nitrogen bases can be either of two types. Either they can be purines or they can be pyrimidines. Purines are adenine and guanine. Pyrimidines are CTU, that is cytosine, thymine, and uracil. You can see the structure over here. Pentose sugars in nucleic acids they are mainly two types. Either they can be ribosed in RNA, or they can be deoxyribosed in DNA, or they can be deoxyribosed in DNA. Now you can see over here, this is the structure of the ribose sugar, where at the second carbon, there is only hydrogen, no oxygen. It is ribose. Sorry, it is deoxyribose. And if there is oxygen at the second carbon, we call it as a ribose sugar. We call it as a ribose sugar. So you can see over here there is OH. So this is ribose sugar, and this is only H. So it is deoxyribose sugar. Everything else remains the same. Then there is phosphate group, which is inorganic phosphate molecule PO4 as we call it. So usually in a nucleotide, the sugar is bound to the phosphorus and to the nitrogen base, as you can see over here. Okay, so as you can see, this is how the nucleotide is there. So let's say this is the just a moment, better just a moment. Over here, these are purines, these are pyrimidines. When it is bound to the sugar, this is sugar as you see, pentose sugar. This is also uracil bound to the sugar, then you get the nucleosides. And when the phosphorus is bound, you will get the nucleotide. So this is nitrogen base, this is sugar, and this is phosphorus. So that is how you get adenylate acid. So these are different nucleotides and nucleosides. Like adenine, you will have adenosine. Nucleotide is adenylic acid. Guanine, you have guanosine. Nucleotide is guanilic acid. Thymine, you have thymidine. Nucleotide is dihamidic acid. Uracil, you have uridine. Nucleotide is uridylic acid. Cytosine, you have cytidine. Nucleotide is cytidylic acid. It is very important. Okay, frequently they ask this. So we have in DNA we only have ATCG and deoxyribose sugar. RNA we only have AUCG. DNA doesn't have uracil, RNA doesn't have thymine. Okay, so this is the basics about the nucleic acid. Any issue in this, beta?
No, sir.
Okay, so now we are sharing next thing. So now we are discussing about the bonds which we see. Okay, about the bonds which we see in this. So there are, if you take two nucleotides, okay, that is, let's say this is the first nucleotide. This is the first nucleotide. This is the second nucleotide. Then the bond is mainly, this is what we call as the phosphodiester bond. So this phosphorus, okay, this three carbon of one sugar is linked to 5 carbon of another sugar through the, okay, and that forms the phosphodiester bond. So between phosphate and sugar is what we call as ester bond, and as there is one such bond, we call it phosphodiester bond. So these are linked to one another by phosphodiester bond. This is glycosidic linkage, nitrogen base and sugar. This part. At 12th માં બી હું 11th માં જ કરાવી દઉં છું 12th માં પણ. And then when this phosphorus is bound through this ester bond, this nitrogen base over here, it is phosphoester linkage. It is phosphoester linkage. As you can see, it is linked to the OH hydroxyl group of the sugar. Now, if you talk about this structure of, like, if you take the DNA structure, okay, for example, let's say we take two nucleotides. Okay, so this is the old diagram. This is one nucleotide sugar, then there is nitrogen base, and then there is phosphorus. So the linkage is over here. This is the phosphodiester bond. Same over here, as you can see. Okay, nitrogen base are inside over here, and two strands of DNA are anti-parallel. One is in this direction, other is in this direction. You can also see over here, you'll get a better picture that for example, over here, if you see, you'll get to know that this is what we call as sugar, phosphorus, and nitrogen base. Okay, these are bound to by this through this phosphodiester bond to the next nucleotide. You can see these are also nucleotide, and you can see over here nitrogen bases. This is adenine is there. Sorry, this is thymine is there over here. Okay, as you can see, there is thymine, then there is adenine. Over here, there is guanine, there is cytosine. There are two double bonds, there are triple bonds over here. These are this is in this direction, and this is in this direction. So two nucleotides are linked by 3,5 phosphodiester bond. As you can see, this is phosphodiester linkage. એટલો important નથી, but you should know. So if you talk about the polymer of nucleotide, that is polynucleotide, then 5' end has free 5' end of the sugar with free phosphate molecule, and over here there is 3' end, there is free hydroxyl at the 3' end. So you always refer a nucleic acid strand or nucleotide strand by 5' to 3' polarity. Okay, so if you see, these are sugar, phosphorus, sugar linked to one another. This is sugar, phosphorus, sugar, sugar, phosphorus, sugar. So that forms the backbone. On the inside is a 12 diagram. Inside we have A and T, which is double bond. Okay, G and C, which is triple hydrogen bond. You can see the hydrogen bonds. So we are going to discuss about B form of DNA, that is secondary form. It's very important. It also comes in 12th. So it is also termed as Watson and Crick model. So it is a double helix. You can see two helical strands over here. Both the strands are anti-parallel. One is 5' to 3' direction, as you can see. Okay, other is 3' to 5'. So these are anti-parallel. The backbone is formed by sugar, phosphate, sugar. This is the sugar, phosphate, sugar, or if you analyze it. Inside there are perpendicularly arranged nitrogen bases. So nitrogen bases are over here, like AT, GC are over here. So these are perpendicularly arranged to this. A always forms 2 hydrogen bond with T, and G with 3 hydrogen bond with C, as you might know. As you can see, always the bonding is AT and GC. So each helical strand appears like a helical staircase, as you can see. If you separate both them, then they look like this helical staircase. જૂના ઘરમાં જે gold stair હોય ને સીડી એના જેવું. Now, as you go above at each step of ascent, that means if you go from here to here or here to here, it is represented by base pair. So this is one base pair, this is another base pair, ATGC, as we can call. Now there are two small calculations that if you want to calculate about length, then between two base pair, the difference is 3.4 angstrom. So the total length of one helical strand is 34 angstrom or 3.4 nanometer. So there are 10 steps in this. In B form, there are 10 base pair. If you count this, these are 10. See, I have mentioned over here. So it is 34 divided by 10. So then you will get distance between one full ton as 10 base pair. So rise per base pair is 3.4 angstrom. Total 34 angstrom length. The base pair distance 3.4 angstrom. Also, if you want to calculate the angle, one full ton is 360 degree. Okay, so you divide the 360 by 10, so you'll get rise per base pair will be 36 degree. Okay, so this is each of the DNA distance you should know, and rise per base pair you should know. So this is about beta nucleic acid. It is very important. 11th 12th બંનેમાં આવે છે and you should know about this.